this is where all the beauty comes in. This is our whole story. Arrangement in space of all atoms in our chain. Again, it's not always clear where secondary ends and tertiary begins, but I'm going to make it clear for this class. Secondary ends when we're only talking about hydrogen bonding in the backbone, and tertiary begins when we talk about interactions of R groups, of our side chains. Those are groups that we classified in the last exam as polar, nonpolar, acidic, or basic. Quaternary is much the same as tertiary if we have different subunits that come together, the same interactions. Now, based on their three-dimensional structure, we have two types of proteins based on the shape that results in their tertiary structure. A fibrous protein that has the shape of a fiber, and globular proteins that have the shape of a glob. All right, let's talk about fibers first because they don't really do much other than give mechanical strength. They're long fibers or sheets. They're quite strong, and they usually are insoluble in water. So because of this, they're structural in nature. Keratin and wool, collagen of connective tissue in animals. So I think that I need to take this little treatment. But now you can get shampoos with it that are supposed to do that. I don't know if it would ever. She needs to stop dyeing her hair. That's my advice to her. That's a problem. But I think a few years ago you could go to the hairdresser, right, and get a keratin treatment. Would it really do this to your hair? Would it make that look like this?
Styling does that to you.
Styling does that to you. I don't know if that would do this to Dolly. But that's the keratin, and obviously it gives mechanical strength. It's fiber. All right, but globular ones, that is what we're very interested in. Again, here's my structure of protein tubulin. This is where a anti-cancer drug would bind. However, notice the shape is not really spherical. That's why we call it globular. More or less spherical. No, not really. Just a glob. All right. They tend to be soluble in water. Not always. The bigger they get, the less soluble they are. But what happens is, because they're soluble in water, mostly, that we're going to fold it. And so the nonpolar side chains are buried inside. And this is an entropy-driven force. And this is the strongest force or the strongest drive in folding of proteins, this entropy, trying to get rid of the nonpolar waxy side chains from the aqueous environment. We don't want to have that ordered chain around those nonpolar side chains. We want to have those buried inside. But there are a lot of interactions that come together when we are folding our proteins. Notice with tubulin you have, here's an alpha helix. Oh, there are some beta sheets. Some beta sheets thrown in there. So, but then it stops. When you see the strings, that's when there's no secondary structure there. All right, so you have an alpha helix, and for some reason it stops. And then there's a beta sheet. It looks like maybe there's a beta meander or something going on there. And, so it really does come along and form different groups. But I do want to point out that that you do have portions of your protein that aren't involved in beta helixes or alpha beta sheets or alpha helixes. Most proteins do have some, but it's not the whole story. So here's a nice comparison of our fibrous and globular protein. Globular looks like, again, a glob. But remember, it does have this three-dimensional shape. So we're going to spend some time here talking about the forces. And again, as you're condensing your notes here, what I would do is go through the levels of your protein structure, define them, and really talk about the forces involved in each. So again, let's summarize. The primary structure, what's the force in the primary structure? Peptide bond. Secondary structure, you have alpha helix and beta sheet, and the force is the hydrogen bonding in the peptide bond. So in the backbone, I mean, in the peptide backbone. So when you define, if you don't say hydrogen bond in the peptide backbone, points off, right? We don't want points off for that. So, and then the tertiary structure, we're looking at the interaction between the side chains. So that's important. Now, see, the reason why in fibrous proteins the secondary structure will give us most of the story is because we don't really have a folding. It's fibrous. It's one long line. So the secondary structure does give most of the tertiary structure information. So we're going to see how things fold back on each other in a globular protein. All right, so the forces that are involved, and you got to know all of these. This is all side chain interactions. So let's think about our side chains. You might want to even look back in your notes on where I looked at those R groups, the 20 amino acids. Serine and threonine both have a hydroxy group. So they would have hydrogen bonding between each other. There's other ones that will have hydrogen bonding as well, but these are just examples. You have hydrophobic interactions between nonpolar side chains. Valine and isoleucine have nonpolar side chains. So those are going to interact. Why is that jumping? You guys see that jumping, or is it just me? It's jumping. Maybe people are walking around upstairs. Now this is, we have electrostatic attraction between side chains of opposite charge. So if you have a basic one that's going to be positively charged at neutral pH or a physiological pH, and an acidic one, like glutamic acid, that's going to be negatively charged at physiological pH, then those are going to form what we call a salt bridge. So there's a couple things I want you to know. Let's stick at this electrostatic attraction for a second. Our basic side chains are going to have a higher pKa. And because of that higher pKa, at pH 7.4 is the pH of blood. What's the pH inside our cell? About the same, right? Somewhere around neutral, right? It's going to be positively charged. So below the pKa for our basic side chain, it's going to have proton on, which is going to make that nitrogen a quaternary amine, and it's going to be a positive charge. So summarize, basic side chains, positive charge. In cellular conditions. Acidic side chains have a low pKa. So when we're at neutral pH, we're going to be above the pKa, and the proton will be off. So it will be negatively charged. So if we have a positively charged basic side chain and a negatively charged acidic side chain, they're going to be attracted, cation to anion. Now, if we put these acidic and basic side chains on the outside of our protein, what's going to surround the positive and the negative? Water molecules. Around the positive, the oxygen of the water molecule is going to surround it. They're going to be hydrated. Around the acidic side chain, the positive side of water is going to be around it. So where do you see these electrostatic attractions between the side chains of opposite charge? Are they going to be out on the outside of our protein or on the inside? On the inside. Has to be away from the water for salt bridges. So this another term for this electrostatic attraction between side chains of opposite charge, that's called the salt bridge. You're going to hear me talking about that, salt bridge. Now, similarly, if you have two basic side chains that are positively charged or two acidic side chains that are negatively charged together, they're going to repel each other. So they're going to try to stay away from each other. Then there's one covalent interaction, and that's where we are going to oxidize two sulfurs on the two different cysteine residues. An amino acid, one amino acid, we call it amino acid residue. I don't know why. It's an amino acid residues. They are going to oxidize. We're going to get rid of the two hydrogens, and there's going to be a covalent bond. So two cysteines can come together and form a covalent bond. Did anybody ever do a gel electrophoresis? You know, did you use beta-mercaptoethanol, some smelly stuff? No, they don't do that anymore, I don't think. Okay, so you're going to have to add, when you do SDS, gel electrophoresis, what you do is you add soap to it, and you also have to break those sulfide bonds. And so that is breaking. When you have a perm, what you do is you put the curlers in your hair, and then you break all of the disulfide bonds in your hair, and then you add another chemical to add them back in. But it adds it back in the form that the curler is in. I'm a child of the 80s, so I'm very familiar with perms. Men would often get perms with very long hair in the 80s. The music was great in the 80s, but the fashion was not. I look back and it's...
Bell bottoms are great, though.
Bell bottoms were not part of the 80s.
Well, that was the 70s.
That was the 70s.
What about parachute pants?
It was high waters, actually, in the 80s. We actually would roll up our pants. It was not a good thing. So you guys need to know all of those, but here's a nice little summary. So here, this is our alpha helix and our beta sheet. I don't know why they do this as a ladder. These are supposed to be the red dots, just like we showed the hydrogen bonds here. So that's not it. But these are not the tertiary structure, but we are going to look at the interactions between these. So here, we're showing that hydrophobic interaction where our nonpolar side chains are going to go into a little pocket. Here's some hydrogen bonding between a hydroxy group and maybe an acidic group. Doesn't really matter. Anything that can do hydrogen bonding. Here's a salt bridge between a positively charged basic side chain and a negatively charged acidic side chain. Here's our disulfide bond between two cysteine amino acids. We can have some metal coordination we'll talk about later. But here's, it's just a nice little example. So I'm going to go through a couple more slides and then we'll stop for today. So in our quest to understand how proteins morph, we often want to know the exact three-dimensional shape because we can't just take a look at the amino acid sequence and say, hey, it's going to fold like that. We can take a guess, and we'll talk about that later, but we don't know for sure. So we take an X-ray of our protein. Now, there's some downsides to this, that you have to have a protein that's a crystal, that's a solid. When proteins are native, they're in an aqueous situation. So not always are we going to be able to use a crystal. And this is when I was in grad school, a big bone of contention in the tubulin world, where someone had come up with an X-ray of tubulin and said, hey, this is our tertiary structure, where we know tubulin was big. If it was in a solid form, it was not going to be in its native form. So they came out with this structure, and my group that I was researching with disagreed with them. We said, we don't believe that when you make a crystal of tubulin that it's going to be in its native conformation. But it is an X-ray, but what happens is because it's on a molecular level, what happens is they pass the X-rays through, and based on the interactions of light, remember when you pass light through and it comes back and it forms diffraction pattern? You get that diffraction pattern, and you have to do a Fourier transform on it, and you can't do it. You get some, and I'll show it to you in the next page, but you need a computer to get the information off of it. What is more modern is to do a 2D NMR, and those of you that have come from RPI, they have NMR that is strong enough to be able to do it. In fact, I used to take my analytical students over to RPI to see the NMR instruments that you would have to climb on a ladder in order to go down and put the sample into it. Anybody come from RPI? Anybody graduate from RPI? No. But my contact there did not get tenure because he was too good of a teacher. I would bring my students over, and he would explain NMR in a way that I've never heard in such simplicity. I could never repeat. He was so good. But because he was too good of a teacher, they had to get rid of him. So he no longer is there. So I don't bring my students there anymore. But NMR looks at the chemical environment around carbons and hydrogens. For those of you who have never heard of that before. But what I like about it is that it can be, and it's in natural environment in aqueous solutions. So here's the diffraction pattern that gives us the tertiary structure of a crystal, and this is 2D NMR. From this, we can look at the structure of our protein. I know that's very complicated, but when this is your life's work, you will get some information out of this. But yeah, this is wicked hard to do. One-dimensional NMR is hard enough for those of you that had organic. Two-dimensional, yeah, I got into in grad school, but this is with proteins, you really have to have an expert helping you out. Structure and the interactions that were involved in each. So we know that there's 4 levels of protein structure. Primary is the amino acid sequence, and we have the covalent peptide bonds, which are also known as amide bonds. Putting those together. We have secondary structure, which are only a local 3-dimensional structure, meaning not the whole story. And those are alpha sheets and alpha helix and beta sheets. And those are interactions in the backbone. Hydrogen bonding between the hydrogen that is covalently bound to the nitrogen and the oxygen on the carbonyl, bless you. And so those are the 2 structures that are involved in the secondary structure. Tertiary structure, we have the involvement of the R group, and we have some non-covalent interactions between those. So we have our hydrogen bonding, hydrophobic interactions, electrostatic, meaning charge attractions and repulsions. And then we have also one covalent interaction called the disulfide bond between 2 cysteine amino acid residues. The last level is quaternary. The quaternary structure is when you have more than one subunit, and those subunits come together and interact in the same way that the tertiary structure interacts. So you have R group involvement. We ended up talking about how we determine the tertiary structure. We can try to predict how a protein is going to fold based on its amino acid sequence, but it's not always going to be accurate. So we'll have to rely on either X-ray crystallography or in more modern times, and a much more complex technique is the 2D NMR. Myoglobin was the first protein for which X-ray crystallography was used to determine the structure. It is a great example, very simple example of a tertiary structure. And many of you know the function, know a lot about myoglobin, but can you tell me what the function of myoglobin is? Stores oxygen where? In the muscle, that's right. It's only a single subunit, pretty short, 153 amino acids, and it has what we call a prosthetic group. So let me talk about what that means. A prosthetic group is just like a prosthesis on a person. It's something that's non-human that you add to a human to make them whole, right? To make them functional. So here we have a non-protein heme group. The heme group is our prosthetic group that attaches onto our protein to make it whole. We're going to talk a lot about a heme group later on, but really what the heme group is the part that makes the myoglobin active, and it does the binding of the oxygen. A lot of aqueous environment proteins have a lot of alpha helix. This one does not have any beta sheets. And again, the polar side chains are on the surface to interact with water, and non-polar side chains, the R groups, are folded into the interior. Now, at the site where the heme group is involved, we have two histidine side chains, and they're involved with the interaction with the heme group and the oxygen. So the heme group has what we have coordination sites. We're going to talk about that in a moment, what does to coordinate mean. But the 6 sites, 4 interact with nitrogen atoms on the heme, 1 with a nitrogen on histidine, and 1 with either an oxygen molecule or on a nitrogen on another, what we call gate histidine. So here's the structure of myoglobin. You can see in the red we have where the heme group is, and then the green are just the amino acids. And I want you to notice that scientists have named the regions of the myoglobin structure. So here's the N-terminus and here's the C-terminus. And we might say, oh, there's a glycine in the GH region, and so people will know what you're talking about. So they can kind of visualize. People that spend their entire lives thinking about myoglobin, they'll and there's people that do. My research advisor spends her entire life thinking about tubulin. So, there's people that spend their lives thinking about one single protein. So you can see that we've labeled them letters. So just be familiar. You don't have to be familiar with this one, but just know that that's what that means. These letters is just so that people have a little shortcut. Oh, we're in the FG region here, so that they can do that. Let's talk about this heme group. This heme group is iron 2 with 4 5-membered rings, and they're called protoporphyrins. They're pyro-like rings. You guys that haven't taken organic don't need to know that, but just know that they're ring structures around. The blue are nitrogens. So the blue ones here are nitrogens. And let's talk about what it means to coordination chemistry, or to covalently coordinate with the iron. It's hard to say. Iron is in the transition metals, right? And what orbitals do transition metals have access to that other metals in, say, group 1 and 2 do not, in the upper part of the periodic table? Come on, chemist. One chemist in here. D orbitals, right? D orbitals, right? and there's some empty d orbitals. So what a coordination bond is when both electrons come from the nitrogen and rest in those empty d orbitals. So there's a lone pair on each one of these nitrogens, and the lone pair is resting inside an empty d orbital on the iron. So here, these silver bonds here, the electrons aren't shared. They're not coming from the iron. The iron still has a 2+ charge, but what it is they're just residing in, they're like freeloading in the empty d orbitals. And so here, this part down here, this comes from the myoglobin. So this is sort of like an anchor. And this coordinate covalent bond stays there. On this histidine E7, this one sometimes coordinates with the iron, but when oxygen's there, it doesn't. So what I like to think is this is, the coordinate covalent bond are like fangs that kind of hook on to our iron and hold it in there, hold it in place. So the fangs come from the snake, not from whatever the snake is going into, right? So the nitrogens have the fangs, and it kind of just grabs onto that iron. So I do expect you to know what a coordinate covalent bond, because we're going to have a lot of coordination going on. Metal ions are important. So this iron, when it's not coordinating with this histidine, will coordinate with oxygen. Oxygen has a lot of lone pairs, so the oxygen will come down and coordinate with it. When it doesn't have oxygen, it will go up with this histidine. So this histidine E7 is sort of like a gate. Now, only when myoglobin has this nice structure, is folded in this nice way, in this particular way, is it native. And only when it has the heme is it active. But some conditions cause denaturation, where we're losing our quaternary or our tertiary and sometimes our secondary structure that will give the loss of biological activity. One thing I want to make sure is that you understand that we are not degrading the protein. Degrading means that we lose the primary structure. We still have the peptide chain intact. When we denature, we still have the peptide chain intact, but we're losing the biological activity. So we lose the biological activity by losing the quaternary structure, if it has it, the tertiary structure, and then sometimes the alpha helix and beta sheets will also get lost, but not always. But by losing that tertiary structure, you're going to lose your biological activity. Sometimes we want to denature, sometimes we don't. When I was in industry, I would like to hire chemists, but the problem with chemists is that they would not know how to work with proteins, because some proteins, if you look at them wrong, they will denature. I would like to hire biologists, because they would know how to work with proteins, but then they wouldn't know the chemistry that was needed. So it was very difficult to find the right person to work in my lab. So I would just have to teach. And then I realized, oh, I really like to teach. And I came here. So see how life works. So one thing that for sure causes denaturation is heat. You guys all right? Can I answer a question? Is it? No. Okay. Interrupt me if you guys are behind on something or need a little extra clarification. I welcome it. It slows me down, so that would be a good thing. So heat. Let's think about, what is heat doing? When you heat something up, what are you doing to the molecule? You're causing an increase in what kind of energy? Kinetic or movement, right? If you're moving, what can we do? We might break those intermolecular forces that are holding it together. So all those intermolecular forces that we listed for the tertiary structure could be broken. Maybe not the disulfide bonds, but you might break the other ones. So heat causes vibration. So I will need you guys to know not only the definition of denaturation, but also what causes denaturation and why. So again, the definition of denaturation is the loss of biological activity through the loss of our structure. We're not missing that primary structure. Primary structure is still intact. If we lose that primary structure, it's called degradation. So heat causes vibration, which breaks our intermolecular forces. Large changes in pH. Remember, if we're changing pH, we can change the charge on our acidic or basic side chains. So if we are having a negatively charged carboxyl group at neutral pH when we're above the pKa, if we go really low, we might protonate it. And so it's uncharged. This is supposed to be NH3, and this is supposed to be NH2. Sorry for the question marks. I know them. So here at physiological pH, this is going to be NH3 with a positive charge. But if we go very high in pH, this is going to be NH2. So what kind of interaction would large changes in pH cause? What attraction or repulsion? I call them what kind of bridges?
Salt.
Salt bridges. So large changes in pH can affect our salt bridges. Can also affect our electrostatic repulsions. The next one is soap. SDS is soap. It's a detergent. Have any of you, I may have asked this question, done SDS gel electrophoresis? Maybe? No? Some of you? When you must have seen them on TV, at least, where they have the gel and the blue bands on that. So in that technique, what we are doing is we're separating based on size. Now, if we have the protein and it's folded up, fetal sort of position, all folded up nice in a nice little tight ball, then we're not really separating just on size, we're separating on shape. So when we want to separate just on size, we unfold the protein and then run it through this mesh of gel. And the big proteins get caught up early in the gel, and the small ones can travel further down. And then you stain it with something that stains to proteins, and you see those nice little bands. Some of you must have. You've done it, okay. And so if you've done the non-native one, the SDS gels, what you're doing is you're adding soap so that just like you get soap out of your soap to use to get the grease off your hand, what we're doing is SDS has a long tail and a polar head. That long tail finds its way into the hydrophobic parts of the protein. And then it makes that polar side of it. So it makes the non-polar parts of our protein polar, so it unfolds. Remember I said that hydrophobic interactions are the main driving force for the folding of proteins. So by adding soap, what we're doing is that hydrophobic tail makes its way into the interior of our protein and has that polar head on it. Does anybody need any further explanation on that? So then just like if I had a little piece of grease on my hand and I put soap on it, soap has a long hydrophobic tail, hydrocarbon tail, that dissolves itself into the grease on my hand. And then what we see on the surface of the grease is just the charged part of the soap. And then so the water can wash it away. And that's the same thing with the detergent, such as SDS. So normally we want this to happen when we're doing this technique, so detergents will disrupt the protein. Urea or guanidine is really a neat thing. Urea, guanidine, we call them chaotropic agents, C-H-A-O tropic. I like to think of it as chaos causing. And these chaotropic agents disrupt hydrogen bonding. These molecules, what they they can do is do a lot of hydrogen bonding. So CHAO tropic. So it's CHAO tropic. And so they will hydrogen bond. They'll go around and hydrogen bond in the place of things that should be hydrogen bonding with other things. So they butt themselves in to where maybe two polar amino acid side chains should be hydrogen bonding with each other. These guys go in there and disrupt that hydrogen bonding. And in the old days, running a gel used to be a smelly job because we would not only want to denature our protein, but we would also, in order to completely denature, we would add not only SDS, but mercaptoethanol. And what that does is it reduces a disulfide bond. And this stuff is smelly. I think they have new chemicals that aren't so smelly because it was really a smelly job. This is a nice little picture of what's happening. Here's our active, correctly folded native protein, and here's our denatured protein. But look, there's a double arrow. What does that mean? You can go back. We can refold if you're nice. Okay? There's some points that there's no going back, but you can refold it. And I can tell you here what we're doing here is adding to say this is ribonuclease. What we're adding is urea and beta-mercaptoethanol, and here we get the denatured reduced protein. And then what we do is sort of do dialysis, and we allow, you remove the urea through dialysis, and then you allow it to air oxidize, and it will pop back in. Now, this only works for small proteins. You have a large one, it's going to be really hard to do this. Not to say that it can't. Yes?